Our surroundings influence our actions. What do I mean by this? Well, let me explain. In order to be a functional adult, we have to pay bills, respond to emails, fuel our cars, occasionally eat and drink, and find time to do things we're passionate about. This means we need to stay on top of all of the information emitted from our environment. Bank details, emails, restaurant recommendations, and meetings. Our phones receive all of this information, and we process it. So how do cells stay on top of their bills? Well, the same way we do. Located within bacteria and human cells are molecules made of protein called receptors. By receiving information about the cell's environment, they're able to help the cell in responding to change, just as our phones alert us about changes through notifications. In cells, receptors receive information in the form of signal molecules known as ligands. We'll be focusing on the one with the fancy abbreviation, TGF-alpha, short for transforming growth factor alpha. TGF-A is a protein. A quick reminder, a gene is a segment of DNA that carries the instructions used to fabricate proteins. In order for DNA to turn into protein, a couple of intermediate steps need to take place. Keep this in mind. Every cell in your body contains DNA containing instructions that fabricate certain proteins. Here's an example of 3 types of cells in your body that produce TGF-A. When TGF-A finds a specific receptor located on the cell's membrane, it binds and gets activated. Both proteins are complementary, meaning a specific ligand will only bind to a certain type of receptor, like a dynamic puzzle, allowing the cell to distinguish between different signals. Right, so our protein TGF-A has found its matching receptor and is currently activated. What happens now? A biochemical cascade. As intimidating as this figure might seem, all it is telling us is that when one thing happens, another one will happen as a result, and another one because of the previous one. You get the idea. Eventually, this series of cellular events leads to a response. But how? Through the addition or removal of a phosphate group which either activates or deactivates proteins. In this example, not all components have been shown for the sake of simplicity. The idea behind this biochemical cascade is to produce proteins such as ELK1 and STAT, seen here, that bind onto DNA and trigger the production of very specific proteins that will bring about a very specific response. One of these functions is cell proliferation, an increase in number of cells. This can be really useful. Angiogenesis, the growth of new blood vessels for example, increases the amount of oxygen that reaches injured tissue and speeds up wound healing. Uncontrolled cell growth, however, is exactly what happens in the case of cancer, leading to the formation of tumors. Unfortunately, angiogenesis in this case provides the tumor with oxygen and nutrients, encouraging it to grow and spread. As you can see, cell signaling can be a double-edged sword. In order to maintain the correct balance, these biochemical cascades need to be tightly regulated. Reactions are inhibited by, you guessed correctly, more proteins. These proteins deactivate components downstream of the signaling cascade, preventing the final response. Responses, be it in the micro or macro range, mostly manifest themselves as detectable events, which scientists can observe and study. And this is what they've been doing for quite some time, gathering information about how different pathways lead to specific responses. But what if we could modify these pathways? What if we can optimize a pathway so that it gives us a desired response? Well, we can, potentially. Modifying the blueprints of proteins allow us to influence pathways. Currently, this is being done with small organisms such as bacteria, but these changes could have significant impacts on a larger scale. Naturally, these possibilities need to be considered with precaution. As previously mentioned, these pathways are very complex, and modifying one for a specific purpose will end up influencing another, possibly unknown pathway in an equal unpredictable manner. Signal transduction is complex, but essentially builds up on the simple principle of transforming an external signal into a cellular response. This video is only possible due to our generous sponsors. We'd like to take the opportunity to thank the innovative manufacturing GRP for their support. Thanks for watching. If you like this video, check out the other videos on our channel.